It's 12:32. So I think it's about time we could start. We have a very good audience. So welcome everyone to the GTP seminar series. This is the second one for this semester. Rob Woods is gonna be the presenter today. And he's gonna talk about the role of spike protein glycosylation in COVID-19, the most, I guess, relevant topic. So we are all looking forward to hear about it. Thanks, Rob, for accepting the invitation and giving it.
My pleasure. Hello, everybody. Thanks for the free pizza. That was awesome. Like you say, hopefully we can meet again in person in the not too distant future. Yep. The title, I realize is the role, but it really should be plural. There are many roles of glycosylation, so we'll cover a few of them today. I divided the talk roughly into two parts. The first is discussing the impact of glycosylation on immune recognition and response to the virus. And the second is the impact of glycosylation of the receptor and the spike protein on adhesion and in principle infection. And then I'll touch on some of the most recent data we have on experimental values for the impact of glycosylation. A pretty picture here is one that shows the spike protein in gray interacting with the ACE2 receptor, and the stick structures that I call moss are glycans. And we'll come back to that soon. So here's a typical representation of the life cycle of SARS, or indeed many viruses. And what you'll see is that the slide presents adhesion as the first step, then endocytosis, replication, and finally budding. What they leave out in this is what's going on here in terms of glycosylation, which everybody in this audience understands is quite important. And this is what I'm going to focus on today. Before we get to SARS, though, there have been a lot of other viruses studied. And one of the things we can learn from this is really how glycosylation varies in general among viruses. And what we see here are hemagglutinins, which you could call the spike protein, if you will, from influenza A, various subtypes showing the glycosylation picture positions as a picture with yellow balls being the glycans, varying as a function of the year or the strain of the hemagglutinins. And the reason I put this up is from studies like this, we've learned that changes in either the density of the glycan, so the number of glycocytes, or their positions on the surface can alter the virulence of the virus and can attenuate the efficacy of vaccines. So in cases even where the protein is relatively constant, the virus will, through mutation of glycosylation sequences, either add or delete glycans from its surface. And that changes the protein parts that can be recognized by antibodies. And so we would like to model this sort of thing because we believe that glycosylation can impact the ability of antibodies to recognize proteins. And the challenge from our point of view really comes down to how do we integrate the data that we get from a glycomics study into a model like this. And it sounds straightforward until you look a little more at detail. So here's a case in point. Again, sticking with influenza, here we can see just at one glycocyte, all the different glycans that were observed when the virus was expressed in express SF plus cell line. And here's another cell line and another and another. And these are all cell lines that are commonly used to express vaccine constructs. And from a glycobiology standpoint, what you can see is that insect cells and mammalian cells often give rise to very different glycans. You can see these structures here are what we might call posse-mannose, where it's just man 2 or 3, something like this. Whereas on the end here, where we've got HEK cells, we get things that are multi-antennary, sialated. And so this is all a fascinating observation, and it depends on the nature of the protein, depending on whether it's more highly processed or less highly processed. It depends on the position of the glycosite. Is it highly accessible to enzymes or inaccessible to enzymes? And it depends on the flux through the Golgi. So one of the challenges we face is when somebody says, well, give me a model of the glycoprotein. My first question is, well, which glycoform do you want us to model? Because if we count and say maybe there's about 10 different glycans that can be on any given site, if we've got n sites, that's 10 to the n different glycoforms. And people largely don't think about this, right? I mean, you just think, well, there's a bunch of glycans, and so model it. And I flip it back to the mass spec guy and say, well, okay, which glycan do you want me to model at each site? And it creates a very interesting dialogue. If we think further about the typical kinds of data you get from proteomics, glycoproteomics analysis, you get, yeah.
I have to interfere and ask a question, of course. Can you go back?
Yeah.
So this is a very interesting, I mean, we know that, but is the fact, if you look at HCK 293, and you have all these different glycan modification in site 1, is this by one typical experiment or different people, different HEK cell? Can you tell a little bit more about it?
This was a study where they expressed the same HA or the same virus in different cell lines. And it's just one person's experiment.
Okay.
And these are all the glycans that they observed, or at least the masses that they observed.
Okay, so it's not different people, different HEK cells, different conditions.
No.
Okay. Thanks.
Right. And I just said these are the masses they observed, and that's really quite important because that gets to this slide here. When you get a glycoprofile, right, it's a bunch of different glycan structures, and if you're lucky, they've been done in a glycoproteomics kind of way, and you can say, okay, at site 293, each of these 5 glycans were observed. And if you're even more lucky, lucky from a modeling perspective, the experimentalist has reported what they consider to be the most dominant glycoform. So for example, it might be this MAN 5 structure. Yeah. But the thing that's missing from all of this is any sort of linkage information. We don't know, are these gal beta 1,3s, gal beta 1,4s? Is this for example, if you had sialic acid, or this is Lewis X, is it? Or is it a different Lewis antigen? And the reason this matters is because glycans have 3D structure, and that's the very basis of modeling. And 3D structure depends on a knowledge of the linkages. So this is the basis of a long-standing debate between some factions in the mass spec world who say, you know, you got to pay attention to linkage, and some who say, no, you don't, because we know it doesn't really vary among mammalian glycans. And there's some truth to that, right? I mean, we know it's always going to be a beta 1,4 glycnec, alpha 3, alpha 6 man. We know this. Out here, though, for example, you don't know because it's rarely examined. Is it alpha 6 or alpha 2, 3? So you could make an assumption based on the cell type. For instance, if it's CHO cells or HEK cells. But there are cases where you've got mixed 2,3 and 2,6. And when you don't have that information, you have to make assumptions. When you put it together, this is how the 3D looks. We draw these as branches as if they're pointing up in the air like the arms of somebody surrendering, but in fact, that's not really the case. Here's the katabios core, and one arm is up and the other arm tends to be either up or down, right? Okay. Once you've decided what the glycan sequence is going to be, we then come back to the question of what glycan do you want me to put at each site for modeling? The most abundant in a given class, or does it even matter? And this latter one is quite interesting. We looked at this because from a modeling perspective, if we have to follow the glycomics data, then we can't be predictive, right? We're always waiting for the data to be reported. And maybe depending on the feature you're interested in, maybe you could just say, I'm just going to put MAN9 at every site. Because you know MAN9 had to have been there at some point, right? As the progenitor glycan, it was there at some time. So maybe it's representative of a pre-processed state. So we take, if we want to model spike or any other glycoprotein, we generate or search for a structure of the protein. We might go to crystallography. That's the #1 place to turn. We might turn to AlphaFold now that it's flavor of the day for protein structures. And then you've chosen what structure of the glycan you want and you put it on. Okay. And what we've shown here is just different ways of representing the same thing. This is gal in yellow, glycnac in blue, man in green, and so forth. Here we've colored according to the terminal glycan. So this one terminates in sialic acid, which is purple, so the whole glycan's purple. And this gives you a quick way to look that this is sialic acid, this is gal, this is high man, and so forth. Now, this is great for generating an image for a paper. You can do this with no big struggle. But everybody in the audience has said or heard that glycans are flexible. And this, of course, is just a single shape. So one of the first questions that arises is how do we capture the impact of motion? And here's a study where they were looking at the core glycans in an FC domain and using a technique called MD simulation, which we'll get to in the next slide or two. And what they showed is that the glycans have different levels of flexibility, even though they have the same sequence. And so this one, you can see the core, glycnac man, is really well defined. Whereas here, it looks pretty flexible. So not only are glycans flexible, but where they are in the protein, they may be more or less flexible. Relative to a protein, if we think about the scales of motion, glycans move approximately on the same scale of motion as amino acid side chains. So the backbone of a protein is relatively rigid compared to the motion of a glycan. So let's take a little detour into how we deal with motion. And the first way we're going to do it is we're going to approach it with physics. And we're going to use a couple of equations. And most of these equations are ones that you've all seen. This is one you used today. You use it every day. You use it so often you don't even realize you're using it. I was at lunch at position X, let's say at Burger King at some time T. I drove at, say, 30 miles an hour for 15 minutes to get to my new position, which is here to give a lecture. How do I know it's a new position? Well, it's this time, 30 minutes, time to get where you were. So you start here, sometime later, you're at a new position. Perhaps I accelerated from time to time and so forth. So we use this all the time to predict where you're going to be if you're starting in a certain position and you're traveling at a certain velocity. And we're going to use exactly the same thing to predict the motion of atoms. So instead of thinking about miles per hour and hours of travel or minutes of travel, we'll be talking about angstroms in terms of distance and femtoseconds or something like that in terms of atomic motion. And we can rewrite that in a simple way to get rid of velocity, which gives us this equation. Okay, so there's the first thing. We can predict where an atom's going to go if we know its current position, its previous position, and its acceleration. And this is exactly what your car's GPS uses to predict where your next turn is coming, where you're going, how long it's going to take you to get to Atlanta, all that sort of thing. We need a couple of other things. We need to know how to get the accelerations. And everybody memorized F equals ma, right? But unlike this equation, nobody ever uses F equals ma. It's a totally useless equation for day-to-day life, but we all know it. And it says if you know the forces on an atom, you can calculate the atomic accelerations, or if you know the forces on an apple or on a jet or anything else, and you know the mass. Well, you can rearrange this, which is another equation, work equals force times distance, which people have heard of, used in your first year physics and then forgot. But it lets us come down to this, that the acceleration is equal to the change in potential energy, or the change in work, divided by the change in distance. And this we get from a force field. So classical modeling is often described as using a force field. And a force field is this big equation down here, which will not be on the exam, but it encapsulates all the hand-wavy descriptions that we as chemists and biochemists use to talk about the relative stability of a complex. It talks about the energy from bond stretching or angle bending, the internal rotational energies, or van der Waals repulsions or attractions or electrostatics, hydrogen bonds, all these things that we would say, why is this shape of the molecule more stable than that shape, is captured mathematically like this, which gives us a way to calculate the energy of the molecule. From that, we can get acceleration and predict position. From a new position, we get a new energy, new acceleration, new position, and this cycle keeps going 1 billion, 10 billion, 1 trillion times, kind of like the national debt, right? There's no limit to it. We can let this go and go and go. And what we get out of this is what's called a trajectory. It's a model, like a video, of the motion of a molecule. To make it accurate, we add water and we heat it to room temperature. And from that, we get something like this. So this is our spike protein. We started off with the glycans in the position that we arbitrarily added them in. And now we've warmed it up to room temperature, surrounded it with water and counterions, and let it move. And that is great for a video, but not so great for a publication, right? I mean, until we get something like the Hogwarts Times, we can't really put a video in a publication, although people do this with a QR code. Instead, we have to have some other way of displaying the motion. And that's what you see on the right. This is what we call, what I call a moss plot, because it looks like the sugar is bunches of moss. And what we've done here is we've just overlaid all the different shapes of a given glycan on top of the shape of the protein. And so you can see that this blue glycan here, for example, covers this much area over the course of a simulation. The high mannose structure here is a little smaller, well, perhaps it's a posse mannose or something, covers less space. The reason we do this is that this ensemble, this video, this collection of shapes is an objective model, whereas any given shape that we arbitrarily put the glycans in to start with is subjective. This is now objective, and it lets us calculate average properties. So we could calculate, for example, the average area of the protein that was exposed to recognition by an antibody in the presence or absence of a particular glycan. And that's exactly what we do or did for this particular work. Now, the question is, how do you find the area exposed to an antibody? How do you define that? Well, we said, being, physicists first assume it's spherical. And actually a spherical definition of an antibody variable domain isn't so bad. You can see 4 antibodies here. The variable domains are superimposed on a pink colored sphere, and we can then adjust the radius of the sphere to be approximately big enough to model the size of the variable domain of an antibody. Now, some loops, some of the hypervariable loops, like here or here or here, these by their very nature are variable. And so we can have a smaller probe that's able to model the loops. So what we do with this now is we roll this ball all over the surface of this, and we see where it touches the protein, and we say there it's exposed and in principle accessible by an antibody. This lets us then take that video and calculate regions on the surface that are accessible. And so that's what you see on the right. Here's a spike protein with no glycans attached. The most accessible regions are shown in red. The most inaccessible are shown in black. It's perfectly obvious when you look at it, but this isn't just visually accessible. This is accessible to a ball the size of an antibody. Now we've done the same thing in the presence of the glycans, and you can see that the glycans actually appear to obscure a large part of the surface, leaving only this receptor domain up here exposed, and the domain around the equator, and a domain down here at the protease cleavage site. So from this, in principle, we can then say, hey, I bet we can predict where antibodies bind. And if we do that, we would predict that they would bind. This is looking down at the top, this is a trimer. So you can see the threefold symmetry. This is the receptor binding domain, one of the 3 in the up conformation, which we'll come back to in a minute. And you can see it's exposed. It's not shielded by glycans. And here's the protease cleavage site. Okay. So this suggests that if you were going to raise antibodies against spike, well, where would they probably target? Well, they wouldn't target the carbohydrate region, likely. They would target the protein exposed region, which is this equatorial band around here, this region here, or the receptor binding domain. Now, when we first started this project, there was no glycomics data. Subsequently, Max Crispin published data. We worked with Lance on data collection on this. Parastu published data. So in a big flurry, a lot of data became available, but initially none was available. And so we said, well, let's model this. We knew where the glycosylation sites were. Let's model it with all man 9. And then we said, well, let's take a hypothetical complex structure or a core-fucosylated complex or a posse-mannose structure and calculate the antibody accessibility for each one. And then we can compare it to the non-glycosylated protein. Later, when we got the glycomics data, that's the HEK data here, we could recompute it using the most abundant glycan at each site. What was interesting that came out of this was actually the result isn't very sensitive to the glycan. Of course, with the smallest glycan, it makes a big difference, right? This is a tiny glycan, doesn't shield, only shields about 30% of the protein. So 70% is exposed to antibody recognition. But all of the others, and it's not terribly surprising, they're roughly the same volume. They shield it approximately equally well. This is quite appealing from our point of view because it means we can go ahead and make some predictions in anticipation of getting the glycomics data. So now let's look in terms of sequence. And what I've done is I've mapped out all the glycosylation sites in a protomer. They're marked with the stars, and you can see each one is on an NXT or NXS glycosylation site. And the color is now the shielding by antibody recognition in the presence or absence of the sugar. And so the darker the blue, the more shielded the site is. So not surprisingly, almost every time when there's a glycan attached, the residues in the immediate proximity are dark blue, meaning they're obscured from binding, obscured from recognition by an antibody. But there are also large regions that appear to be obscured from binding because of the presence of sugars, but that are not directly in contact with the glycans. So this is the case of, say, the head group of the glycan, the end branches shielding the surface, as opposed to here where it's the core of the glycan shielding the surface. Now, how do we know this is real, right? How can we tie this with anything other than modeling? And so when we first published this, there were a few structures of the novel coronavirus in complex with antibody, monoclonal antibody fragments, and there were a lot of complexes of related coronaviruses, for example, from SARS or MERS. And you can see what I've done now is I've overlaid the structure of glycosylated spike on the structure of SARS or MERS and shown where the antibodies exist that recognize either SARS or SARS-CoV-2 or MERS. And you can see in all cases, neutralizing antibodies tend to bind to the receptor binding domain. Now this is sort of satisfying because we predict that that's the most exposed region. But there are a couple of things that come out of this. If you look very carefully at this structure, you will see that there's an antibody in this pale blue transparent structure that seems to collide with a bunch of sugars. So what's going on there, right? Does that mean that our model is wrong, that the crystal structure is wrong? What's going on? The other thing to note is why aren't there antibodies in co-complex with these things where the antibody's not on the receptor binding domain? Why aren't there antibodies down here where in all cases the protein is exposed? And I'll put it to you that that's for the reason that the effort that goes into studying these is focused on neutralizing antibodies, right? Antibodies that block infection or kill the virus. So if they're going to block infection, they have to be blocking the receptor. That doesn't mean there aren't antibodies that recognize down here. It's just they're not of biological interest because they don't do anything in terms of the infection. So why does anybody study those? Well, in general, they don't, because all the interest is in the antibodies that modulate infection. So there probably are many antibodies that could interact down here that just aren't studied. Now, looking back at this structure here, where there's a mass of glycans that appear to collide with an antibody. Well, we zoomed in on this, and here's the antibody in pale cyan, and the crystal structure has the glycan at that site over here. Whereas we show it as a big mass or moss of glycan. And I think the point to remember is that when we do this, what we're presenting, what we're showing in that image are all the different possible shapes that that glycan can adopt. But it doesn't simultaneously adopt all of them. So for example, during the MD, there's a shape that we see here in pink that's actually quite similar to the one in the crystal structure. And the pink one allows the antibody to bind. So what this means is when we're looking at these MOS plots, they give a visual aid to showing how much of the surface of the protein is potentially shielded, but they overestimate that because at any one time, only one shape is adopted. So the averages we get, the sequence plots where we color it in different shades of blue, that's accurate because that's based on the average behavior. But the visual of having a bunch of moss on the surface, that overemphasizes the apparent shielding of the protein. So if we now compare antibody epitopes that are known, which are in green, so this is this plot where we've now shown the sequences that are shielded or in contact with an antibody. They're shown in green for MERS, SARS, and coronavirus, CoV-2, versus our predicted shielding. You see a really good correlation where we predicted would be exposed for antibody binding. That's where antibodies are known to bind. So that was fairly satisfying because that means that we can now go forward and predict how a particular mutation or a loss or introduction of glycosylation might impact an epitope. So going back to this idea where we know that glycans move around season to season, and this is why we're very likely going to have to continue to take booster shots as new variants come out, especially as they move glycans around. We can predict the impact of glycosylation on antigenicity. That is, we can predict whether or not a current vaccine is likely to be effective against a given variant.
Hey Rob, is there a trend that the glycosylation is increasing over the years on these viral surface proteins? I'm just asking as a naive question. From like 1918, there seems to be very little glycosylation as part of the virulence mechanism, I guess.
Yeah, people have said that. I think generally it's believed to be the case. And as you can imagine, there's a competing evolutionary pressure. The virus wants to shield itself fully from immune surveillance. But if it puts too many glycans up in the head group, then it can no longer bind to the host. So it has to leave its receptor binding domain exposed. Otherwise, it loses antigenicity. And in fact, this is what happened in the H3s, right? The H3s put so much glycan up in their head group that you stop being able to detect them in typical binding assays that were geared towards the receptor.
Thanks.
So there have been many, many studies. I mean, this field is just, wow, exploded. But here's some data that talks about known variants. And some of these involve glycans, so I've highlighted them. N149 to Q and N149 to H both knock out the glycan at 149. N165Q knocks out the glycan at 165. And T719A knocks out the glycan at T721. So if you then look at the impact on antibody binding, you can see that the loss of either of these two glycans actually led to an increase in binding to convalescent sera. So it would appear that these two glycans were shielding an epitope that became more exposed when they were removed. And if we look at where those two glycans are, remember 149 and 165, they're up here in the head group, up here in the receptor binding domain. So their loss, we show that they shield the neighboring amino acids quite a bit, right? So this glycan N165, there's a lot of blue around it, meaning it's shielding all these residues. If you knock it out, these residues now become more accessible for antibody binding. In contrast, down here, it seems to have no effect.
How do you distinguish between adaptability, binding to the receptor rather than to the immune system, to escape the immune system?
Well, this is just binding to convalescent sera, right?
Mm.
So it's not immunization.
Yes, but but changing of the amino acids, like you show in the previous slide, could be adapted to a better way to bind the receptor, correct?
Yeah, so you're right. So I'm going to get to that in the last couple of slides, because you're absolutely right. There's an adaptive immune response that's totally been ignored up until this part of my talk.
Oh, thanks.
And here it is. So for those who don't know, when we get an adaptive immune response, what we're looking for is presentation of peptide antigens in an MHC2 or MHC1 complex on the cell surface. And this triggers T cell response and builds memory cells and so forth, right? Now, whenever non-glycobiologists, virologists, and biochemists talk about this, they're always talking about a peptide antigen, with the exception of FRSC. Because
some other people do too, but yeah, me included.
He is one of a small group of people, and it isn't, I mean, it's a growing group, largely because of the work he's done pioneering this concept, that the peptide could be a glycopeptide, right? So there's nothing that says you couldn't have a glycan attached to this peptide, except that, I mean, even if you just look at this simple cartoon, how do you fit a glycan in? And, you know, FRSC back in 2011 in a really groundbreaking publication said, well, you know, if the glycan's small enough, maybe it doesn't matter. And as somebody else said, well, this is sort of, I know it shows it like a ball, but it's really a linear binding site. If the glycan's at the end of it, perhaps it doesn't matter. So the potential exists then for glycosylation to modulate adaptive immune response, largely in a negative sense, by blocking the ability of this T cell recognition to occur. But it doesn't have to block. So then we went through and we said, all right, let's look at the sequence from another light. There are servers out there that'll predict which peptide sequences are likely to be expressed as in an MHC2 complex. And those are ones that I've put a rectangle around. And of those ones, the only ones I looked at were the ones that also contained a glycosylation site. So this is a putative HLA antigen, as is this and this and this. And if you, to get back to the earlier question from Maor, if you delete a glycan here, it very possibly makes this more amenable to presentation in an MHC complex, and therefore leads to a better immune response. That wasn't what the data was for, though. That was convalescent sera, but it could still be that, right? And in fact, there was, and I meant to put it in the talk and I forgot, but there was a paper where they studied the immune response to a nude glycoprotein, right? So it a spike without glycans, and they got a really good protective immune response. So I think generally the glycans aren't doing anything good for immune response. So the conclusions from this so far then are the glycans shield about 40% of the spike surface and that is relatively insensitive to the glycan type. Some epitopes are impacted more than others based on where the glycan is on the surface. The receptor binding domain, not surprisingly, is not shielded, because if it were, it could potentially block the ability of the virus to bind to host. Glycan microheterogeneity, though, that's a different thing. This is now, we're talking about, imagine you got 10 different glycans possible at each site. Well, the distribution of glycan microheterogeneity depends on many factors: cell age, organismal age, underlying disease, and in humans, ethnicity. So microheterogeneity likely impacts differential responses to the virus. The innate immune response that I didn't get around to discussing, but these are lectins that bind to the glycans. That will be highly impacted by microheterogeneity. And of course, this could affect the adaptive immune response. But what did we get out of this first part? Well, we got the ability to define and predict how different glycans affect antibody recognition, and then you can begin to probe for differential susceptibility as a function of microheterogeneity. So the shorter second part of the talk, what I want to do is just discuss the interaction with ACE2. I'm showing this image because it shows in the trimer, this one receptor domain is up. There's actually an equilibrium between up and down shapes, and it is the up shape that interacts with ACE2. And here we see ACE2. ACE2 is also a glycoprotein. It's this red blob here, or the color, the white translucent blob here, and the glycans, the 6 glycosites are as shown. And you can see that there are some glycosites, notably N322 and N90, where the glycan is just about, well, it's essentially at the interface of the ACE2 and spike. And a lot has been made of this. Here's an MD simulation of the co-complex. And what I've shown in pink are the glycans attached to ACE, and then gray, the glycans attached to spike. And in blue surfaces, the glycans that are attached to ACE that appear to interact directly with the surface, like this. You can see that no matter how you look at it, there's an interaction there for sure. Of course, we don't know whether it's favorable or unfavorable, it just is, yeah? So we wanted to probe this and try and get a grip on what's going on. So zooming in, yeah.
So do you assume that at the interface of binding, there will be no salic acid from both glycans, the ACE2 and the spike protein, or it doesn't matter?
There are some of them are sialylated, yeah. Yep. And 165, the dominant structure is sialylated.
So you wouldn't assume that the ACE receptor protein will have sialylated glycan as well, right? Or
well, I know we think of these things as if they're repulsive interactions, right? Because it's two negatives. Is that where you're going?
Well, you know, it's a modeling. So how is it coming into your calculation when you're planning this docking type of?
Well, this is a co-crystal structure. This is no docking.
Okay.
All we've done is add the glycans that are observed from glycomics.
Mm-hmm. Okay.
And so you can see this one makes a pretty good contact from ACE2 with the spike. That's N322. N90 also makes a contact also with a glycan from spike, and N546 is just glycan. So here's a case of glycan with protein and glycan with protein only.
So you have about 4 or 5 amino acid contact and 2 or 3 glycan contacts. Is that?
Yeah, something like that.
Okay.
So we got Kelly to express some protein and Lance, and we took our fancy schmancy biolayer interferometer and made some measurements. And first of all, we just got a sample that had been deglycosylated. And if you deglycosylate ACE and you bind it to wild type spike, you get about a 5-fold enhanced affinity, 4-fold. And if you deglycosylate spike, but you don't deglycosylate ACE2, so you keep ACE2 wild type, it gets worse. So it either gets 4 times better or 4 times worse, depending on what you deglycosylate. So yes, there's a role for glycans. Now, if we start to pick this apart a little bit more, we now have some point mutations, and we can say, well, this N322 that's buried deep in the core here of the interface, if you remove it, it binds about 2.5-fold tighter. So this is largely an unfavorable interaction. And the N90S binds also about 2-fold tighter if you remove this glycan. 546 over here had essentially no impact on binding. And this comes back to this up and down receptor binding site. So if we think about just ACE, we said, well, we didn't know whether these were repulsive or attractive, but they're clearly jammed into the binding site. Just by saying jammed in makes you think they're unfavorable. And in fact, that's what the data says. But removing the glycans on spike, it's not so obvious why they would impact it. And it turns out that there's been a lot of work, largely coming from cryo-EM and modeling, that showed that the up conformation is modulated by the presence of glycan N165, that this glycan bolsters the up conformation. So when you don't have a glycan here, you get more of the down or closed conformation and less ability to bind. So in light of that, it makes perfect sense that when you deglycosylate spike, it attenuates binding. So in some cases, the glycans indirectly help binding by pushing the receptor binding domain up into a favorable orientation. And in some cases, they hurt binding just because the way the recognition has evolved, they're in the interface. So to conclude, what have we got? Well, microheterogeneity is going to turn out to be really important for understanding the differences in individual susceptibilities to infection and to infection by variants through breakout infections and that sort of thing. What did 3D modeling give us? Well, it helps to define the molecular basis for all of the biophysical observations. And you can think of now, what does glycosylation do? What are its roles, plural? It impacts vaccine efficacy. It impacts the innate immune response and the adaptive immune response, presumably, and infectivity due to differences in ACE2 spike affinity. As is noted here, that certain mutations in ACE2 increase viral entry, right? So there are some polymorphisms in human ACE2 where mutations are likely to make people more susceptible. Okay, so to close this, this is really a large collaboration among both several groups of experimentalists from outside my team and experimentalists within our team and modeling people. If you're interested, you can download these models for using in publications or using for your own data analysis at this site. And that's what I had to talk about.
Thank you, Rob. It was a lot of learning and great structures. So we'll take some questions.
I have a question, Robert. Very nice, interesting talk. So it has been said that actually the receptor for the ACE2 is one of there are other receptors in the spike protein for other protein. I think the N-terminal domain of the spike protein is also involved in some sort of binding. Can you show where it is located, if you know about it, where it is located? Is it the exposed surfaces?
Yeah. So if we just look at this structure for a minute, here's the stalk region. This is the membrane surface down here. The other commonly accepted binding partner are glycosaminoglycans, because we and others published and pointed out early on that there's a glycosaminoglycan binding sequence down here. And so it's thought, and I've seen a nice picture of it, that the virus is loosely held onto by relatively non-specific glycosaminoglycan interactions with things like heparin sulfate that hold it long enough to allow it to form the stronger interaction with its direct partner ACE2.
You know, early on, you showed us this very nice cartoon, exposed and unexposed, with the moss covering. Can you show it again, and can you let us know if this is the area that is involving binding? Yes. Oops. Yes, this one.
Yeah, it's somewhere down in here. Yes.
Okay, so these are so-called the predicted exposed surface that may be involved in binding to other receptors in human.
Yeah, most notably to glycosaminoglycans.
Okay.
Hey, Rob, this is Bob Haltewanger. Can you hear me?
Yes, Bob.
Yeah, so you made a really interesting statement in the conclusion about microheterogeneity in individuals that might influence infectivity. Which would, I mean, so have you changed? I mean, you could do subtle changes at any site using modeling. Have you actually done that to kind of see whether it would have that big of an effect, that it would change infectivity?
Well, we haven't decided how best to answer that question. What we did that was related, though, we looked at in flu the binding to a innate immune receptor. I'm trying to remember which one it was, and showed that MD simulations could predict the glycans that were involved in the threefold symmetry recognition of one of the collectins, right? So from that perspective, yes, we could say that you can identify the multimeric partners that are involved in binding to the innate immune receptors, and then predict how changes in their structure would impact that. But it's one of those things that's a little hard to test.
Sure, no, but it's a really interesting idea. So thanks.
Hi, Rob. Great talk. This is Parastu. Can you comment on the fact that for these new mutants that are more virulent, there is no change, none of the glycosylation sites change actually with the mutants like the Delta and the Alpha ones, none of the there is no deletion of any glycosylation site. And there is, in fact, no significant change in glycosylation in the mutants.
Well, as much as I hate to admit it, sometimes it's not all about the glycan, right? But when you look at a distribution like this, for example, if you make a change to an underlying amino acid. It can change the region of the surface that the glycan is covering. If we take a specific case of this, right, this whole region is an area that the glycan spans, but at any given moment, there can be an orientation that allows an antibody to bind. If you make a mutation, for example, over here somewhere that's unrelated to the glycan, it may still impact the shape distribution of glycans in that region, allowing more or less exposure of the surface. So I think what I'm saying is that you can have an indirect effect on the exposed area of the protein. It could be exposed to antibody, it could be exposed to receptor binding, right?
But we can also, but we can also say that they're so critical that the virus is not changing anything because it needs those glycosylation sites. So it's not really changing those sites.
Well, so far, right? Yeah. I mean, think in terms of flu. In one season, how many glycosylation site changes do you get? But next season's a new thing.
Mm. Hey, Rob, it was a great talk. This is Lance.
Thanks.
I had a question on your last slide. You mentioned a recent study showing that the N546 Q cells expressing that ACE2 form are more susceptible to infection. Yeah, right there. So, you know, N546 is interesting in ACE2 because about 2 million people on the planet don't have that in-link glycosylation site. Do you know, has anyone tried to, you know, figure out whether those patients, I mean, based off that data, you might think they're going to have a more severe, you know, course of disease. Do you know if anyone's looked at that?
Well, I was trying to get that together. I knew somebody to ask. I was trying to get that together before the talk and couldn't find it. But
yeah, I've looked too, and I can't find that data. I was just wondering if maybe I'm missing it.
But I think the thing is, it's actually the 548 is the polymorph, which knocks out the glycan. It's an ST polymorph, and I was searching 546. So I think if we focus on 548, you might find it.
Oh, that's a good point. Beautiful talk. Thanks.
Sure, Lance. Thanks for working with us on this. Sure couldn't have done this without you and Kelly.
I guess I'll since you mentioned about MHC2 presentation of glycopeptides, in fact, a group from UK early on in 2020 showed, along with a number, like they did the immunopeptidome of what gets presented with the spike protein, along with several peptides. They have also shown that glycopeptides, truncated glycopeptides, are also presented for T cell recognition. Which pretty much aligned very well with our work on an HIV enveloped glycoprotein, in which we have shown that such glycopeptides with oligomono structures are presented. So that was good because it was kind of validating our HIV work, which they cited. But at the same time, that was the study that made me like, hey, yes, we are one of the few labs that study this, but I'll let somebody else work on the spike protein presentation. So that kind of discouraged me to keep moving on that end. But that was a good work.
You can't do everything, right?
I can't. I actually love when people do things that validate our data. So that's amazing.
So actually, if you think of it, could you please send me that paper?
I will, yeah.
Because I'd like to compare it to this and see where that fits in.
I'm gonna actually just send it to you right now. Any other, yeah, I forget that I was the host. Any other questions for Rob? Well, let's thank him one last time for this great talk, most relevant. And we look forward to seeing you guys in our next GTP seminar, which you will get email reminder for. Thanks again, Rob.
Thanks, Fikri. Bye, everybody.
Bye-bye.